the PCR product which are obtained from a previous PCR mixture, if they are going to be used as a template for any next PCR reaction, in that case we have to use a different method to purify the PCR product. Why we have to do it? This is because of the fact that the PCR product is having unused dNTPs, and if we don't remove these residual or unused dNTPs, they will create problem with the subsequent method. The overabundance of the primers which were used for a specific amplification in the previous PCR mixture can interfere with the next PCR amplification reaction. Therefore, removal of these oligonucleotide primers is mandatory. If we are planning to use this sample for cloning purpose and the primer dimers are present in the PCR product mixture, then these primer dimers can act as template for restriction endonuclease. If they are carrying any restriction site, in that case they are going to be in competition for the original template DNA, and these primer dimers are going to decrease the efficiency of enzymatic action. Therefore, they need removed. Different methods are used in labs to remove or to purify the DNA which is obtained from PCR amplification. Traditional method is use of phenol chloroform, but this method is slow, is messy, and comparatively inefficient. Therefore, we have to find out some alternative method. Another method, the chromatographic method, that uses the anionic silica gel resins or some other specific resins to purify the DNA, are also in practice. The spin dialysis method is most practiced method these days. No doubt it is slow, is less efficient, and is less able to handle many samples at a time. With the organic method, this is possible that we can handle many samples at a time for purification, but in spin dialysis, we are restricted for number of samples to be used for purification. However, despite of these drawbacks, these problems, this method is preferred for the reason that it can give us the reproducibly good quantities of purified DNA, and it can remove the dNTPs and oligonucleotide primers very efficiently. During this method, what we have to do, we are basically going to use a filter paper that is having a specific sized pores, and when the PCR product is applied on that paper, all those solute particles which are larger than the pore size, they are retained, and rest of the things, they are filtered. These macromolecules are then recollected in the desired solvent and can be processed further for any required reaction or the PCR using this previous PCR product as template DNA.